I'm not only going to talk about modeling, but also a little bit about the challenges for experimental structure determination. And the overview is that I'm first going to talk about structure, function, and some families of membrane proteins. Then structure determination with experimental data. And then I'm going to go into the prediction, sequence-based prediction, de novo structure prediction. I'm not really going to talk about homology modeling or fold recognition simply because I could talk about membrane proteins for much longer than an hour. And we simply don't really have the time. I'm going to talk about a little bit about Rosetta membrane, the work that I've been doing with the undergrad that Jeff mentioned, and what we have done, what some of you guys have also used and noticed, and other methods that are out there, and then also future developments. I mean, membrane proteins are very important since they're in every single membrane in the cell, whether it's mitochondrion or nucleus or ribosomes, you know. Every cell organelle has its membrane, and membrane proteins are in there, and so they're extremely important. And also, actually, like a fun fact, scientists have tried to predict membrane protein structures even before the first membrane protein structure was experimentally determined. So this was something that I recently read and was like, that's kind of funny. How do you predict something that you don't even know the structure of? Calcium channel, it's a membrane protein. So membrane proteins are extremely important drug targets, and about 30% of the genome actually encodes for membrane proteins. And over half of all drugs on the market target membrane proteins. So that's an incredible number. So now if you look at structures, if you look at the protein data bank, only about 1% of the structures, currently we have about a little more than 100,000 structures, and about 1 to 2% of those structures belong to membrane proteins. So it's a tiny amount, and then compare that with how important they actually are. Yes?
How difficult is it to transport a drug across the membrane?
Well, you would need methods to do that, whether you're, hijacking a transporter or, yeah.
So most of these aren't going to diffuse across, really?
It depends on what you have, right? If you have a large pore,
okay.
I mean, yeah, some of the drugs could diffuse through that. The question is, whether you have large pores that are open. Typically, the large pores for membrane proteins, this is basically bacterial or viral proteins that make pores into the membrane that actually lyse the cell, so that kill the cell. It's something that you don't really want either. So this is really an ongoing question of research, and I don't know. I don't have a good answer for this.
Yeah, that's part of why these drugs are targeting the GPCRs, because they target
Right,
the cell and set the signal in.
Exactly.
There's also a lot of drugs are kinase inhibitors, right? And those would have to get in. I think one of the drug indices to see if it's a good pharmacological molecule is like hydrophilicity, to see if we can get something diffuse across the membrane. It's definitely one of the barriers. And for complex things in the cell, like gene therapy, that's one of the challenges. You've got to get complex molecular machinery past that.
But for kinase inhibitors, if you're looking at like EGFR, for instance, I mean, these signal across the membrane. So if you target the outer portion and then it can't signal anymore, then you've achieved your breaking down your signaling cascade without actually going through the membrane. But yeah, please, if you have questions, always interrupt me. So. And the reason for why we only have such a small amount of structures determined is that it's extremely difficult to obtain high-resolution structures with experimental methods. For instance, NMR spectroscopy, that's what I've been doing, or what I have done, or X-ray crystallography. And so I will go a little bit more into detail later on. So first, a little bit about structure, function, and families. As you might have noticed, I'm not sure how many membrane proteins you looked at or worked with. Soluble proteins, they can have all kinds of structures, all kinds of angles between helices, all kinds of sheets, looks very messy. If you look at a membrane protein, this is a G protein coupled receptor. Everything looks relatively organized. So the helices have, a certain angle with respect to the membrane. You will never see a helix like this lying in the membrane. You also can have beta barrels. They actually form complete barrels. You will never have unsatisfied hydrogen bonds like on this sheet over here that you have. You will never see this in the membrane. So you basically have two kinds of structures. You have alpha helical bundles like this, or you have beta barrels. And so both of them are much more organized or structured than soluble proteins. So from a modeling perspective, this actually means that it's somewhat easier to model membrane proteins simply because the membrane imposes those kind of restraints onto the protein, and you can use that for structure prediction or folding. From an experimental point of view, that's a bad thing because you actually have to deal with detergents and lipids to make the protein happy and to make it fold properly. So some functions that membrane proteins have, they have important functions like transport, proteins, channels. They have their intercellular junction proteins, and you will see an example later on. Enzymes are sitting in the membrane. You have receptors like GPCRs or signaling molecules that bind something on the outer part and then signal through the membrane and have like a signaling cascade inside the cell. Then you have, for cell-cell recognition, proteins in two different membranes that can bind to each other, enzymes that work as anchoring molecules for cytoskeleton and extracellular matrix. So these are basically one of the main functions that membrane proteins can have. And so if we look at largest families of membrane proteins, these are actually G-protein coupled receptors. And G-protein coupled receptors are actually not only the largest members of membrane proteins, are actually the largest family of proteins. So this is also something that I recently noticed after working on this for 10 years. I didn't know that before.
Is that the numbers in humans?
Say what?
Is that the number in humans? Can't be E. coli or yeast because there's heavy.
I don't think that it's only in humans. I think that's generally the case, but I might want to check. So GPCRs, there's major facilitator proteins, there's transporters, ABC transporters, potassium channels, cytochrome B, you have neurotransporters and channels, glutamate receptors. So this is kind of like the largest 25 families that are out there. And this was from a paper from 2006, so that's 8 years ago. But still, I mean, GPCRs are extremely important. So if you look at channels, for instance, there is channels for passive transport. So, ions or whatever just passes through, the protein doesn't really need to do anything. And then there is active transport or active transporters where you actually need energy to carry your charge or molecule across the membrane. And so if you look at channels, even in a little bit more detail, you can have simple diffusion, that was the passive transport that we just saw earlier. Then you have ion water channels, gated channels, uniporters that only transported in one direction, symporters that transported two molecules in the same direction, antiporters that transported in the other direction, or and then ATP transporters that use ATP to transport things. So this is a huge variety of transporters or channels that you have. And this was just to illustrate how complicated, or this really requires membrane proteins to be pretty complicated. So if we look at largest families in terms of structures, you guys have seen probably GPCRs before. They're kind of look very similar. Ion channels, there's all kinds of different folds that ion channels can have, from two transmembrane spans per chain that form a pore, an oligomerize, to 7 or 6 transmembrane spans. Everything's possible. So these vary in folds. Then you have ABC transporters, so this is like an open and inward open and outward open conformation. So they transport things across the membrane. And then major facilitator proteins like the LEC-Y protein. These are pretty large. I think they have about 12 transmembrane strands total. And so this is just kind of like the largest families that you will come across a lot. So now there's a whole other variety there. These are GPCRs, they all kind of look the same. So these are major facilitators, you've seen this. These are gap junctions proteins. A lot of membrane proteins are symmetric proteins. They look very pretty if you look at them. All kinds of channels and transporters. You have beta barrels that form trimers a lot of times, but not all of them form trimers. So you have, monomers in there. You have pore-forming toxins. So these proteins make holes in your membranes and kill cells. More transporters. These are photosynthetic reaction centers, which I think is probably one of the largest membrane protein structures determined. They're simply huge. And so just to give you like a general idea of how they look like, and they're just, yeah. So experimental structure determination. If you want to determine structures, you're going to run into a lot of problems. The first problem that you will have is overexpression of your protein. So a lot of membrane proteins are actually toxic to the host cell. So your bacteria don't really want to express something that's toxic to them. And so you have to overcome this barrier first. So expression is generally a big problem. Once you have expressed your protein, then you need to worry about a membrane mimetic. So you want your protein structure to be happy, to fold properly. You don't want to have a misfolded or aggregated protein. If you want to determine the structure, you want to have the protein to have a native structure. And you need either lipids or detergents somewhat to keep the protein properly folded. Then if you cross those two hurdles, you probably want to start out with crystallization. The first big question mark is, well, does it crystallize at all? And some people try for years and years and years and never get there. Some proteins may require protein engineering, especially the G protein coupled receptors. They've done some pretty heavy protein engineering to put in, fab fragments or T4 lysozyme insertions to actually make the protein crystallize. And if you can't crystallize it, or some people just do NMR, and for large proteins, there's a size limit for large proteins. So if you want to really go through this will take you a while. And so that's one of the reasons why we have so few structures compared to soluble proteins. So now, for instance, this is the beta-adrenergic receptor. The crystal structure was determined, I think, in 2006 or 2007 after really many years of failed crystallization trials. This took, 15 years until they finally crystallized the protein. This is actually a structure they determined in my old lab after actually 13 years. This is an NMR structure, and I believe this is still the largest NMR structure of an alpha helical membrane protein determined so far. And so it can really take a very long time. The last one, this is a crystal structure. This was just published last year. Can you give me a number of crystallization trials you can try? Say what?
160,000.
160,000. I'm sure they had robots for that, but still, that's incredible. All right. So if you want to determine membrane protein structures, you need a membrane mimetic, and a lot of people are using either detergents or lipids to do that. Detergents have a single fatty acid chain, and if you throw them into your sample, that will form a micelle. This looks like an ice cream cone, and this will form like a round micelle around the protein. So for this also might mean that your protein might have some distortions if you determine the structure eventually. And I'm not going to go into any more detail with that. And so the other option is to use actual lipids, which have 2 fatty acid chains, and you can do like bicels, for instance, if you're doing NMR spectroscopy, so you have more like a flat kind of part of the membrane, and then you close up those ends over here. And so if you have micelles, you can determine structures like this, but you can have distortions from the micelle simply because the micelle is round. So you actually try to avoid that. If you're doing an NMR spectroscopy, these are easier to do simply because they're smaller, and so which means your line shape will be sharper. But you might have distortions. People now typically use bicelles or nanodiscs for structure determination. And so for instance, if you have, and the reason why it's really difficult to do NMR is because if you have small molecules, not small molecules, if you have small proteins that tumble really fast, you will have very sharp line shapes. If you have a really huge complex that tumbles very slowly, you will have a really, low signal to noise ratio and a really broad line shape. And so if you're looking at this from like an actual spectrum view, so if you have soluble proteins and you acquire a spectrum in, let's say, 30 minutes, these are both spectrum that I acquired a long time ago. After about 30 minutes, you see a beautiful spectrum, very sharp peaks, you can distinguish them very well, similar peak intensities, large dispersion, beautiful. For membrane proteins, you run this for 12 hours and you get something like this. So this makes it really way more difficult to, first of all, assign your spectrum, get the constraints for structure determination, and actually calculate the structure. So this is really way more difficult than if you have soluble proteins. So the next thing is also, for NMR spectroscopy, people typically use NOE restraints, and these restraints are distance restraints that you can measure within 5 angstroms. So if, for instance, you're looking at a beta barrel protein, you can measure those 5 angstrom distances across 2 different strands in a sheet, for instance. If you have an alpha helical protein, those NOE distances are only along the helices. You cannot connect those helices with respect to each other, which makes it extremely difficult to determine the overall fold of the protein. And so what you really need to do in this case, you need side chain assignments in your spectra to measure restraints and then use those restraints. But for membrane proteins, this is extremely difficult, if not impossible, to do. And this is also the reason, one of the reasons that for larger proteins, there are more NMR structures of beta barrels, simply because these are easier to determine than NMR structures of alpha helical proteins for membrane proteins. So that's why we need to do computational structure prediction. And the first thing that you typically start out with is sequence-based prediction. And as you know, probably we typically start out with secondary structure prediction. And so in terms of secondary structure prediction, this is not simply only for membrane proteins, but applies as much for soluble proteins. Those methods have emerged sometime in the 90s, at the same time when the first structures were determined. And those methods were pretty much random at that point. So the first generation techniques correlated single residue statistics with secondary structure. Again, they were pretty much random. The second generation methods got statistics from longer segments. They were typically, 17 to 21 residues, and so that worked a tiny bit better. And the breakthrough really came with multiple sequence alignment combined with machine learning techniques. And so machine learning techniques, I'm not sure you might be familiar with it or have heard about it. It's really, basically you can train a computer to predict something. And you train a computer on a database, and it works extremely well. So these methods are still used today. And for secondary structure prediction, for instance, Cypred is a really good predictor. PCL Bluefo9D, this is actually the program that I wrote when I was in grad school, and JPred, and there's tons of other secondary structure prediction techniques out there. So, and this is just a schematic of an artificial neural network as an example of machine learning techniques, but then there's support vector machines and decision trees and all kinds of other things. So for transmembrane span prediction, what is a really important starting point is actually free energies. And you guys have talked earlier on like DDGs of binding. This is not DDGs of binding, but this is basically DDGs of moving an amino acid from solution into the membrane and asking, is it happy in solution or is it happy in the membrane? And so it's really the free energy the amino acid has in solution interface on the membrane, and the lowest indicates preference for that region. So these hydrophobicity scales can either, there's various hydrophobicity scales out there, Wimbley and White, for instance, Kite Doolittle, Goldman, Engelmann, Steitz. Then there's a couple of scales out there, and they can either be derived experimentally, they can be knowledge-based, or they can, use different scales and combine them together in consensus scales. So if you do that experimentally, what people have done is they measure partitioning energies of, let's say, host guest peptides between octanol or into octanol and cyclohexane. So they get experimental numbers out of this, and for instance, for charged residues over here, residues that have peptide bonds or aromatic residues. So you will get different numbers for all of your 20 amino acids. And recently, 2 scales have been derived for fully folded proteins, so these are experimental scales. The biological hydrophobicity scale from Stephen White and von Heine, or the sidechain hydrophobicity scale from Karen Fleming here at Hopkins. These 2 were actually a pretty big deal in the membrane community. So now, if you have those, yes.
How does the free energy and the hydrophobicity scales relate to each other? Are they the same?
They're the same, yeah. So now you can use those free energies to predict transmembrane spans. So if you know, for instance, that your residue will be very happy in the membrane, you can, look for those. You can map those numbers onto your protein, and you will see what, which parts of the protein will be most likely in the membrane or not. And so typically what people have done is they summed those free energies over a window, typically between 10 to 25 residues, and looked for, what they call hydropathy index, for instance. And this scheme actually worked pretty well, and I'm sure some people are still using it, even though there's much better techniques out there nowadays. At least they've used that until, I don't know, 10 years ago. And so prediction accuracies are, below 60% if you use that simple window averaging scheme. And so basically what you do is you run a window over this, and you sum up your hydrophobicity values, and whenever it's above a certain threshold, you say, oh, that could be a transmembrane span, that could be a transmembrane span, and so on and so forth. And that works pretty well for helical proteins, but it doesn't really work for beta barrels. For instance, if you look through a beta barrel over here, you will see that these residues all face the lipid, whereas these guys actually face the interior, which can be polar. And so this wouldn't really work, because if you sum these up, they just sum up to 0. And so your prediction accuracies are actually pretty bad for these. And so what people have done is, again, they used machine learning techniques or artificial intelligence techniques to predict those. And just as an example, since this was also something that I've done in grad school, this is a potassium channel, casease A, and outer membrane protein W. these are the simple hydrophobicity values mapped onto the structure. And you can't really map out like, okay, where is the solution, where is the membrane in here? If you now average those hydrophobicity values, you can kind of like map out where those, the red ones are supposed to be in the membrane, the blue ones are supposed to be outside of the membrane. It works reasonably well, but it's not great. And so if you do that over a database, you get about 57% accuracy. If you now use artificial neural networks, for instance, you can train the method, you know exactly where the membrane is. So this is really, and you get, for a 3-state prediction, whether it's in the membrane, interface, or solution, you get accuracies much higher than this, so it's like 73% or something like that. So also those methods are mostly specialized for either alpha helical bundles or beta barrels. So there's, to my knowledge, only the one that I developed out there that can deal with both types of proteins. Other methods are like octopus, TMMOD, TMHMM. They're all helical prediction methods, whereas Bactibus, beta rare, TM beta net, and so on, therefore for beta barrels. So if you feed in a helical bundle into a beta barrel method, it will, show you like, oh, there's no transmembrane span predicted, even though it's you just. So now structure prediction. Structure prediction is actually, there's like mostly 4 big methods out there. The first one is homology modeling that you guys have probably talked about in class. Then there's fold recognition for if you can't really identify a good template. And we will not talk about these today at all. I'm going to talk a little bit about de novo folding or de novo structure prediction. And there's also molecular dynamics, which I'm not going to talk about, and I'm not sure you're going to talk about here in class. Probably not. So de novo prediction of alpha helical bundles, this is actually the most difficult task, simply because if you want to predict the structure of a membrane protein, it's always good if you can have a template. For instance, for homology modeling, you can say, hey, this is my sequence. I can identify a sequence that is similar to my sequence of interest, and then I can map it back and can do homology modeling. For fold recognition, that's a little bit more difficult. You do a sequence structure alignment, you score it, you pick out your best scoring alignment, and can still have an idea of like, this might be the overall fold of the protein. If you do de novo structure prediction, you don't know anything. You have no idea of how the structure looks like. And this is also why I've been going into so much detail about transmembrane span prediction methods, simply because for de novo prediction, that's the only idea that you have of how the protein might look like. And so there are several methods out there. Rosetta membrane was the earliest method, which was developed in 2006, 2007. And I'm going to talk a little bit more into, I will go into a little bit more detail. And then these other methods have really recently emerged in the past couple of years. BCL MP-Fold, which uses complete secondary structure elements and a moveset for sampling. So using complete secondary structure elements instead of small fragments like Rosetta does, it actually lowers the conformational search space. And so there's also a smart moveset for sampling, and for most membrane proteins and large soluble proteins, it actually currently outperforms Rosetta. Then there's the film 3 method, which also does a fragment assembly. It also uses correlated mutations, which is something that people are starting to use very successfully. And there's EV-fold membrane, which also uses evolutionary constraints. And this method is actually pretty remarkable, at least in the paper that they've shown. If they fold proteins de novo up to 360 residues, they get consistently RMSDs below 5 angstrom. So these are actually all examples from this paper. And so they get the fold right, and they fold it pretty well de novo, which is remarkable. So this is basically just the comparison of those methods. You have the sequence length, and here you have the RMSD to the crystal structure. Typically what you would see, the larger the protein becomes, the less, or the larger the RMSD is going to be. And meaning the worse your protein model will be. And so Rosetta membrane, these are the original, from the original paper, these black dots over here. It does pretty well for, smaller proteins up to like 150 residues. Then you have Rosetta membrane in gray, which is from the BCL MP fold paper that they've used for comparison. And so these get up to 14, 15 angstrom RMSD depending on how long the protein is or how large the protein is. Now, but these don't, BCL, MP fold, and Rosetta membrane don't use evolutionary constraints. So if you now look at the green and the blue dots, they both use evolutionary constraints, which brings down the RMSD to the crystal structure quite a bit. And so this is BCL, MP fold, this is EP fold membrane. So this is consistently below 5 angstrom. This is really a striking result. So now, Rosetta membrane. I just want to talk a little bit about Rosetta membrane. Some people have used it, or at least the new framework in it. And so the energy function of Rosetta membrane is actually split up into layers. So you have an inner hydrophobic layer, outer hydrophobic layer, interface, polar, and a water layer. And so this is the low resolution scoring function. You have an environment term, a pair term, and a density term. So this is really what the low resolution scoring function of Rosetta membrane was implemented, and that is still what it looks like today. So, but the implementation of it in the code, how it was written in 2006, 2007, it all lived in basically 2 classes. And so it was membrane potential and membrane topology. And here in the topology, you would read in your span files and your lipophilicity files, and everything would be basically mixed together in the code. So now, if you go a little bit through the history of this, was in 2006, 2007. Since then, Rosetta was completely rewritten. And they have implemented concepts like a mover that you've probably been working with, confirmation, a full tree. All those new concepts have developed, but this old code didn't really have any access to it at all. And so what we actually wanted to do, and these are the protocols that were supported by the old Rosetta membrane. So relax, ab initio folding, and comparative modeling. And in our case, we wanted to do docking, or if you want to, model symmetric proteins. That wasn't really possible at all with the code the way it was. So we decided to completely start from scratch and think about what we want to do. And so we wanted to develop a new framework that fits several requirements. And so the first thing is, of course, we want to do the science. We want to get the science done. In terms of code design, so I'm just going to put this in here. In terms of science, we wanted to handle multiple chains. We wanted to model large proteins. We didn't want to be like one of the few methods that models membrane proteins pretty badly. And so we really wanted to be able to keep up with the other methods that have been recently developed. And so in terms of the code design, we wanted to use the membrane full tree. We wanted to use the recently developed concepts in Rosetta and have an accessible in-code interface that people can use in PyRosetta or in C++. And also we wanted to improve the user interface to make it easier for the user to actually use Rosetta. And also, of course, develop new protocols with it. So these were our goals that we wanted to do. And this is kind of like the framework that we came up with, pretty much the way it is right now. So the upper part over here is basically what you have access to in a protocol. So if you have your Rosetta protocol, you have access to a scoring function. You will work on your pose, whatever you're doing, on your biomolecule, and you will use movers to manipulate your pose. And we have implemented, for each of those, membrane representations, or not membrane representation, but membrane parts of this. So the score function, the actual scores itself, the actual energy function stayed the same. We just took them out and put them into different parts of the code. So the membrane spores are still there. Then movers, we have implemented a couple of membrane-specific moves that weren't there before. And for the pose, we can now create a membrane pose in Rosetta that you couldn't do before. So I'm not sure how familiar you are with the actual setup of Rosetta, that there's a confirmation object that people can access, and we have put in a membrane info object into confirmation that you can access through. So, which is really important because you can access this object from your protocol level. You can access it from anywhere in the code, which is really convenient because this membrane info object contains all of your membrane-derived information, like lipophilicity or spanning topology and so on and so forth. Then another thing that we've also done is we created a membrane virtual residue. So the way the protein knows about the membrane is really about this virtual residue, which has a center, and it's kind of like depicted here. It has a center atom and it has a normal. And so this is really all the protein needs to know about in terms of, you know, how is it oriented with respect to the membrane. And having this as an virtual residue, which is not present at scoring, it's only there as like a placeholder for us to know, like, oh, this is where the membrane is. We can use that and connect it to the protein in the full tree object. Now, I'm not sure how much you've used the full tree object. This is actually really, really useful because you can manipulate things quite a bit this way. And so that's the basic setup, if you have questions. So really quickly, I don't want to bore you with details. There are a couple of lowest level classes that we have. There's a span class, which basically has the start and the end position of a transmembrane span in your protein sequence. Then there's an embedding definition, which contains the center point and the normal vector of a specific span, for instance. And now, if you have container objects for these, for the spanning topology contains several span objects. So let's say your protein has 2 transmembrane spans. For instance, this is one for glycophrin A. You have start residue 15 to 31 as a transmembrane span, and the second span is from 55 to 73. And for embedding definitions, the container object is the embedding, where you can have, you know, span embeddings, where each span has their own embedding, and you can build things up. You can have chain embedding, which is basically the average of those, and so on and so forth. You can really, things are really quite flexible this way. And so, yes?
If you have a span file, then do you not need to worry about embedding the protein?
So we designed the embedding objects to be able to manipulate things in terms of orientation. It depends on the problem. So there's a couple of problems where you don't really need the embeddings, but then there are also, if you do folding, for instance, it's much easier if you have embeddings for your individual helices, and you can say, oh, these should be embedded this way, and you can manipulate things easier. So it depends on the problem that you have, really. So another question that we also had is like, okay, do we want to have the membrane moving around the protein, or do we want to have a fixed membrane where the proteins are moving in it? And so having a movable membrane is actually computationally cheaper, simply because if you look at your full tree, this would be your protein, and you would have a jump to the membrane residue that would move around. So in terms of pictures, this is how it would look like. You have your protein, and the only thing that really moves around in orientation would be your membrane residue. So in the end, you would only need to update the orientation of this residue, which is computationally much cheaper. On the other hand, sometimes you need a fixed membrane. For instance, if you want to dock two proteins together, you need a fixed membrane that you can have multiple proteins in it, or if you want to. Oh my gosh, okay, I thought it was going until 3:30. Okay, so, or if you want to assemble multiple chains, this is what this is needed for. And so actually you will, if you develop protocols in Rosetta with the membrane framework, you can have both options. You can manipulate things the way you want to, so it's really flexible these days. Then if you want to develop protocols in Rosetta, you need to call the add membrane mover, which sets up the membrane info object, and this is really the first mover that you really need to call. And so what it does, it really adds the virtual membrane residue with the center and the normal in there. And so here are just a couple of application cases that we have. Membrane protein relax protocol. Then we've implemented membrane DDGs, so we can calculate DDG values in the membrane, which correlate, reasonably well. But this is still the energy function from 2006 that will be updated in the next, hopefully, couple months, and not by us, but by our collaborator from us. And we can do docking. Actually, we can dock proteins in the membrane. And so these are really just few application cases that should show you how easy it is to develop things. We started this project last year in 2013 in the summer, and rewriting the membrane framework took pretty much a whole year. My undergrad and myself have worked on this, and since then we've implemented a couple of application cases. There's also, I'm not sure, I don't think I have the symmetric docking here. This is just how docking would work. Like, you would take off the side chains and would go into centroid mode. At the very beginning, you've seen that the membrane residue is added to the pose. Then you would dock it. You would try different conformations. Currently, this chain is fixed, but this will also be movable later on. And at the end of this movie, you will put on the side chains and go into high resolution refinement. This is just like an example of how he puts the side chain X on, and we're not seeing high resolution refinement here. So there's obviously still a lot to do, like plugging your energy function, actually benchmarking it with weight optimization. There's still a lot of work to do, but as a quick and dirty thing, it's very simple. A span file, a PDB file, flags file that has your setup in there, and then you run your np-docking executable. Yes?
When you say that you plug in a new energy function, generally how do you go about thinking about, I guess, what kind of an energy function to design? So do you take actual, experimental measurements for the energy, or you just kind of play around until
So you're asking like a very general question. For this, the energy function that we're using, is based on the old Rosetta membrane energy function that people have derived from a knowledge base of protein structures. But if you're doing like the stuff that you guys did with DDG values, then yeah, you could put in your own values to compare it to the way you have done it. But in terms of really like an actual energy function itself, you would take one that is already set up in the database. So.
Yeah, those centroid scores primarily are knowledge-based. The solvation score that we've been using from Lazarus and CAR plus Lazarus is working with Benoit Roux now, and they're coming up with some implicit membrane solvation models. That's what's plugged in there now, and there's a lot of work in that area. And both on the theory side and experimentally measuring it, there's cleverness to trying to measure what these burial energies are and the interaction area energies in the membrane. So it's evolving.